Welcome back to the Science Channel podcast, where we discuss the coolest scientific papers and preprints of our day. If you're finding these deep dives into cutting-edge research interesting, please take a quick moment to like the video and subscribe to the channel. It really helps us bring you more of this content. Today, we're taking a pretty fascinating journey into the intersection of our environment and our health. We'll be unpacking a study that looks at how something many of us encounter daily, urban air pollution. Specifically, those tiny particulate matter specks.
Right, the really small stuff, PM 2.5.
Exactly, how that can actually increase our risk of developing dangerous blood clots, a condition called thrombosis. And what's particularly compelling here is the exploration of a potential countermeasure. Could metformin,
The diabetes drug.
yeah, the common drug for type 2 diabetes, could it offer some protection against this really harmful effect of air pollution?
It's definitely a really important area of investigation. We know that breathing polluted air is linked to a higher incidence of serious cardiovascular events.
Like heart attacks and strokes.
Exactly. Heart attacks, strokes. It's actually a major contributor to illness and death worldwide. So the idea that we might be able to mitigate one of the underlying mechanisms, especially using a drug that's already well understood, widely available, that's quite exciting, really.
Okay, so let's get into the gritty details then. What exactly is it about this urban particulate matter that makes it so hazardous to our health? Specifically thinking about blood clots here.
Well, what's critical to understand is the specific composition. We're talking about these incredibly tiny particles, smaller than 2.5 micrometers.
Hard to even imagine how small that is.
Right, think microscopic dust. And they're typically composed of a core of, like, ash or carbon. But the key thing is what's on them. They carry condensed organic molecules and heavy metals. Basically remnants from incomplete burning of fossil fuels.
Okay, so stuff from cars, factories.
Precisely. And when we inhale these particles, they go deep into the lungs and come into contact with alveolar macrophages.
Those are the immune cells in the lungs,
Yeah.
right? The cleanup crew.
Yeah, essentially the first line of defense down there. But they seem to have a problematic reaction to this specific type of pollution.
Okay, so what happens?
The research showed that exposure to these urban particulates triggers those alveolar macrophages to release pro-inflammatory signaling molecules, cytokines.
Ah, cytokines. We hear a lot about those in inflammation.
We do, and one cytokine in particular, interleukin-6 or IL-6, seems to play a really key role here.
IL-6, got it.
Right.
So this IL-6 is sort of the central player in this cascade of events. What happens once it's released by these lung immune cells?
So once IL-6 is released there in the lungs, it doesn't just stay put, it enters the bloodstream.
Okay.
And thinking bigger picture, this IL-6 then travels through the blood to the liver.
The liver. Okay, what does it do there?
The study demonstrated that IL-6 actually promotes the liver's production of coagulation factors.
The proteins that make blood clot.
Exactly, the things that help our blood clot when we need it to. But here, you get more of them. So this increased production leads to a higher tendency for arterial thrombosis, that's the formation of blood clots in our arteries. Not good.
Definitely not. And they showed this in.
Well, initially it was shown quite clearly in a mouse model of stroke, but importantly, similar effects were later observed in a human study.
Oh, really?
Yeah, a human intervention trial conducted in China. They found that individuals breathing filtered air showed fewer of these prothrombotic changes compared to those breathing polluted air.
Wow, okay. That really underscores how relevant this is for, actual people living in cities. Now, let's pivot a bit. Metformin. We know it's for diabetes, but what was the thinking? What led researchers to suspect it might help against these pollution-induced clots?
Yeah, that's a great question. It points to metformin's broader biological actions. It does more than just regulate blood sugar. In various preclinical studies, so in labs and animals, it's shown some promising effects, like potentially slowing down tumor growth. Yeah, extending lifespan in some animal models and even reducing cardiovascular risks. So the researchers hypothesized that metformin's known influence on mitochondria.
The cell's powerhouses again.
Right. Specifically, its ability to modulate the production of reactive oxygen species, or ROS.
Okay, ROS. Those are the unstable molecules that can cause damage.
Exactly. They thought maybe metformin, by affecting ROS, might interfere with that inflammatory response that the particulate matter triggers in the lungs.
So the investigation then really zoomed in on how metformin interacts with these alveolar macrophages, the lung immune cells, at a cellular level, especially when they're hit with this urban pollution. What did they actually uncover?
Well, the results were pretty striking. The study found that metformin could indeed prevent that acceleration of thrombosis caused by particulate matter exposure in mice.
Prevent it, like completely.
Remarkably, yes. It brought the time it took for a blood clot to form right back down to the levels they saw in mice breathing clean, filtered air.
That's incredible.
It is. And digging deeper, they found that metformin treatment in the mice led to a reduction in the expression of the IL-6 gene inside those alveolar macrophages.
Meaning less of the instructions to make IL-6.
Precisely. So less IL-6, that key inflammatory driver, was being produced in the first place. And this finding held up both in the live animals exposed to polluted air and in isolated lung cells treated with particulate matter in the lab.
That's a pretty direct link then. Metformin reduces the very molecule, IL-6, that seems to be driving this increased clotting risk.
But, okay, how? How does metformin actually achieve this at the molecular level inside the cells?
Right, this is where the cellular mechanisms get really interesting. The researchers look closely at metformin's impact on the mitochondria within those alveolar macrophages. Remember, mitochondria generate energy, but they're also involved in other signaling. They found metformin significantly reduced the ratio of NAD plus to NADH. NAD plus NADH ratio.
Yeah.
That's an indicator of...
It's a key indicator of the activity of the mitochondrial electron transport chain that's the crucial process for energy generation. So a lower ratio suggests metformin is kind of inhibiting or reducing the activity of this process.
Okay, so it's slowing down some of the mitochondrial machinery. And this connects back to those reactive oxygen species, the ROS.
Exactly. They use a special dye that lights up when mitochondrial ROS are present. And they showed that pre-treating the alveolar macrophages with metformin significantly decreased the amount of ROS produced when the cells were exposed to particulate matter.
Ah, so it seems like metformin is acting quite early.
Yeah.
It's preventing that surge of ROS that the pollution would normally cause inside the mitochondria. Did they pinpoint where in the mitochondria metformin was having this effect?
Yes, they did. They used a really clever technique involving an alveolar macrophage cell line that was engineered.
Engineered how?
They made it express a yeast protein called NDI1. Now, this yeast protein can do a similar job to a part of the mammalian mitochondrial machinery, specifically complex 1. But crucially, NDI1 isn't affected by metformin, and it doesn't produce ROS at that site.
Okay, so it's like a resisted version.
Exactly. And what they found was fascinating. These cells with the yeast NDI1 were resistant to metformin's effects. Metformin couldn't reduce their oxygen consumption, couldn't reduce ROS generation, and couldn't stop the IL-6 production.
So that strongly implicates the mammalian version, complex 1.
Precisely. It strongly suggests that metformin's action on our own complex I is essential for its protective effects against the pollution.
But wait, was complex I itself the main source of the ROS that the pollution was triggering? Or is it more complicated?
That's a really important distinction, and it is a bit more complicated. While metformin acts on complex 1, the experiments showed something else too. If they directly blocked ROS production at a different site, complex 3, that also prevented the PM-induced ROS and IL-6 release. But directly blocking ROS only at complex, I didn't stop the IL-6 release caused by the pollution.
So what does that mean?
It indicates that the particulate matter exposure primarily triggers ROS production downstream of complex 1, likely at complex 3. Metformin, by acting on complex 1, seems to reduce the flow of electrons through the chain, which indirectly lessens the ROS being generated later, at complex 3.
I see. So it's an indirect effect on the problematic ROS source.
It appears so. And interestingly, just using general mitochondrial antioxidants, things that sop up ROS broadly, also provided similar protection.
Right. They also checked another pathway, didn't they? AMPK. Because metformin is known to activate that.
They did look into AMPK activation, yes, but it didn't appear to be the primary way metformin was reducing IL-6 release in this specific context.
Okay, and another subtle point. Just generating mitochondrial ROS wasn't enough on its own to trigger IL-6 release, was it?
Correct. The findings suggest ROS generation acts more like an amplifier. It enhances the effects of the particulate matter on IL-6 release, rather than being the sole trigger itself. You still needed the PM exposure.
Got it. Now, the study also brought in another important player, calcium signaling, specifically something called CRC channels. What are those and how do they fit in?
Right, so CRA channels, calcium release activated channels, When the alveolar macrophages were exposed to the particulate matter, the researchers saw a noticeable increase in calcium ions floating around inside the cell cytoplasm.
Okay, the calcium levels went up.
Yeah, which pointed towards these CRE channels because they are pathways that let calcium flow into cells from outside or from internal stores. And they discovered that metformin actually inhibited this whole calcium response. It inhibited both the initial release of calcium from internal stores and the subsequent influx of calcium through these CRC channels after PM exposure.
And blocking these channels directly also helped.
Yes. When they used specific chemical inhibitors to block the CRAC channels, they also saw a reduction in that PM-induced IL-6 release. It mirrored the metformin effect in that regard.
Okay, so it looks like maybe the ROS somehow leads to this calcium signaling through CRAC channels, and that calcium influx contributes to releasing IL-6.
That seems to be the sequence, yes.
And metformin is interrupting this somewhere upstream.
Exactly. Further experiments suggested that metformin's ability to inhibit the CRAC channel opening happens after its effects on mitochondrial ROS. So by reducing the ROS,
it indirectly prevents the calcium channels from opening as much.
That's the idea. Reduce the ROS, reduce the CRAC channel activation, reduce the calcium influx, and ultimately reduce the IL-6 release.
That's quite a detailed chain of events they mapped out.
It really is.
And to really strengthen these findings, they didn't just rely on cell lines, right? They looked at in vivo evidence in animals and also checked relevance in human cells.
Absolutely crucial steps. They used mice that were genetically engineered. Specifically, they lacked a key mitochondrial protein, TFM, but only in their macrophages.
What does TFM do?
It's essential for mitochondrial DNA transcription and the whole electron transport chain function. So these mice had impaired mitochondrial function just in those key immune cells. And these mice showed reduced PM-induced thrombosis and lower levels of IL-6. It supports the idea that macrophage mitochondria are central to the problem.
Okay, that fits. Any other animal evidence?
Yes. They also treated normal wild-type mice with a specific mitochondrial antioxidant called mitotempo.
Yeah.
That also prevented the development of the PM-induced thrombosis, again pointing fingers at mitochondrial ROS.
And the human connection.
This is really important. They took primary alveolar macrophages, so cells directly from human lungs, and exposed them to particulate matter in the lab. They found that both metformin and a specific CRAC channel inhibitor called Synta66 were effective. They both inhibited the mitochondrial ROS generation, the calcium release, and the IL-6 release.
Wow. So the mechanism seems conserved between the mouse model and actual human lung cells.
It certainly looks that way, which really bolsters the potential relevance.
And it seems the effects of metformin might have gone even beyond just dampening that specific inflammatory pathway. They looked at overall gene activity, too.
They did, using RNA sequencing. This lets you see which genes are being turned on or off. They found metformin significantly altered how the macrophages responded to particulate matter at the gene level in the mice.
In what way?
One notable thing was an increase in the activity of genes that code for chaperone proteins.
Chaperone proteins?
Yeah.
They help other proteins fold correctly, right? Part of stress response.
Exactly. They're involved in things like the unfolded protein response and general cellular stress regulation. They help maintain protein quality control or proteostasis. This suggests metformin might actually be inducing a kind of protective stress response within the cells, making them more resilient.
Huh. That's interesting. And was this linked back to the mitochondria?
Yes, it appeared to be. This upregulation of the protective chaperone genes was dependent on metformin's ability to inhibit complex 1, so it all seems connected back to that initial mitochondrial interaction.
So metformin might have these broader protective effects by sort of tuning how our cells cope with stress. That could have implications way beyond just pollution, couldn't it?
That's a really fascinating possibility, and certainly an area for more research. It highlights that metformin's anti-inflammatory actions and its potential to promote cellular health proteostasis could contribute to its observed benefits in other areas, maybe even aging.
Okay, so let's try and bring this all together. The key findings from this deep dive seem to be metformin, by acting on mitochondrial complex Y.
And reducing the production of harmful reactive oxygen species.
Right, ROS. It can effectively prevent air pollution-induced thrombosis, at least in the mouse model they used.
And it seems to achieve this by stopping the release of that pro-inflammatory molecule, IL-6, from the lung macrophages.
A process that involves calcium signaling through the CRA channels.
Correct, which seems to be downstream of the ROS effect.
And crucially, these protective effects, the reduced ROS, calcium, and IL-6, were also seen in primary human alveolar macrophages.
Yeah, that human cell data is really significant. It underscores the potential clinical importance of these discoveries. I mean, given metformin's safety profile, it's been used by millions for decades, and it's low cost.
It's very affordable.
It really does present a promising potential therapeutic avenue, especially for people at high risk of cardiovascular events who live in polluted areas.
This is truly compelling stuff. I mean, could a common medication for diabetes really hold a key to counteracting the harmful cardiovascular effects of the very air we breathe?
It's a powerful question.
And it makes you wonder, what other surprising benefits might this widely used drug possess, by influencing these fundamental cellular stress responses? Definitely gives you a lot to think about. Please don't forget to like the video and subscribe if you want more content like this.